Hello, everybody. My name is Melanie Parejo. I am a postdoctoral researcher at the University of the Basque Country and specialized on honeybee genomics. I am very lucky and grateful that I received this year the COLOSS Ricola Award for Excellence. So thank you very much to COLOSS Association. And in this mini presentation, I will tell you a little bit more about my awarded project entitled Selection Signatures in an Untreated Surviving Honeybee Population on Unije Island, Croatia. And Unije Island is what you see in this figure in the back. Let's start with a little bit of background on the Unije experiment. So similar to the natural selection experiment on Gotland Island, on the pristine island of Unije in Croatia, honeybees have survived without any treatment since 2012, despite there being Varroa destructor and other pathogens in these colonies. So these colonies are interesting to study because they may allow us to gain insight into the mechanisms enabling colony survival. To investigate these colonies, a collaboration between researchers from Croatia, Germany, and our university started a multi-year experiment that followed the colonies over several seasons, taking different measurements on hive strength and pathogens, and taking various hive samples for microbiota analysis. And then as a comparison group to this pristine Croatian island, the same measurements and samples were also taken in traditionally managed colonies that were set up in Baranja, which is on the mainland Croatia in a typical agricultural landscape. In previous studies, we have already revealed that colonies from Unije Island had a more stable and healthier microbiome with increased beneficial functions compared to colonies in the agricultural area of Baranja. However, it is not quite clear what factor plays the genetic makeup of the honeybees in this adaptation and survival of the colonies on the island. The questions are if there are specific genes of the bees that are under natural selection and that enable colony survival, or is it predominantly the environment that is a driver of survival, or are the key drivers enabling survival the beneficial microbiota that are more present in this pristine environment? So since we already have some data and knowledge on the microbiota part, the aim of this proposal is to look at the genomes of the honeybees on the island. To this end, I aim to compare at the genomic level the colonies from Unije to the colonies in Baranja. So Unije, as I told you, is a pristine landscape with unmanaged and untreated colonies, while Baranja has a typical agricultural landscape with traditionally managed and treated colonies. And you can see, well, the difference of the landscape type in the pictures. The aim is to find selection signatures at the genome level between the honeybees of both populations, and the samples were taken in 2019. From each of the 12 colonies, pools of 10 workers will be sequenced, totaling 120 workers. And whole genome sequencing will be performed using standard library preparation kits and paired end sequenced on an Illumina Novasec platform. Then for the bioinformatic analysis, first, mapping of the sequencing reads onto the current honeybee reference genome will be performed. followed by variant calling and quality filtering using the software population. Then I will perform signatures of selection analysis based on allele frequency differences between the surviving colonies from Unije Island and the managed colonies from the agricultural area in Varroa. And then identify signals in genes involved, putatively involved in survival. Finally, the findings identified at the genomic level, I will contrast them with the findings with already generated microbiota data. So the expected results of these analyses are to gain insights into which genomic features enable colony survival on Unije Island. In particular, what is the relative importance of genetic features of the bees, of the microbiota, or not genetic influence, and is mainly the environment. So you will hear more about these results at the COLOSS conference in 2023, so next year, because I have not yet received the sequence data, and most importantly, I will be on maternity leave very soon, so not able to do the analysis this year. I like to end with this very short presentation with a special thank you to all the collaborators, especially the ones from Croatia, for all the field work and setting up these valuable experiments, namely Janja, Ricola, Marin, Zlatko, Ralph from Germany, Marta, Andone, Irache, and June from Spain. So thank you very much to all of you for this valuable experiments and setting this up. And I'm grateful to now have access to these samples and perform this genomic analysis. And of course, I'm also, again, very thankful to COLOSS Association for awarding me this prize and thereby funding the sequencing of these samples. So it's a great opportunity for me to look into this at the genomic level. I also leave you my email if you have any questions. Thank you very much.